if you do have any, it may, you need some time alone to digest this and review it, but of course, happy to answer your questions anytime on the third floor or by email or what have you. Okay, should we do one leading question? Have you had alcohol or ethanol metabolism anywhere? Yeah, that's needs to be in the curriculum somewhere.
And in a couple spots.
It has? Okay.
Like not, it's been, the photo's been in there.
Okay. It's pretty important medically. Okay, should we just check people's answers? Okay, so acetaldehyde is a product of alcohol dehydrogenase, but it's not a product of fatty acid beta oxidation. The common thing of those two pathways is NADH being made. So they're both oxidative pathways that reduce NAD plus to NADH. Okay, now fatty acid oxidation also makes FAD2H, right? Through the acyl-CoA dehydrogenase step. NADH, the ratio of NAD plus to NADH is an important intracellular indicator of energy status. FAD2H, the ratio of FAD to FAD2H doesn't serve that role. Can anybody think of a reason why?
Does that have to do with like the glucose 3-phosphate shuttle and all that? Or?
No, it's kind of more basic, and it's maybe like the first or second day of the lecture. You have to think back to some cofactors are prosthetic groups of enzymes, and others bind transiently and then dissociate. Okay, FAD2H, that's always associated with an enzyme. So the redox reactions only happen on that protein and only affect that protein. NADH, there's a pool of NAD floating around in the cell, and that can either be NAD plus or NADH. Okay, and that balance is important for a lot of different functions in the cell, and it's particularly important for the TCA cycle. So with the fuel oxidation function of the TCA cycle, it's really driven forward by consumption of ATP and work being done, either transport work or mechanical work or biosynthetic work, what have you. You have to be hydrolyzing a lot of ATP. That then is going to be driving the electron transport chain to be oxidizing a lot of NADH to NAD plus, and then that reduces the amount of NADH for all these reactions, and just by the elimination of the product of the reaction by oxidizing it on the electron transport chain, that's going to drive the energetics of that reaction forward. Does that make sense? Okay, so the regulation of the TCA cycle enzymes, like the regulation of any enzyme activity, is going to be dependent on the concentrations of the substrates and the products, right? Okay, so the key is that NADH is a product of a lot of the reactions in the TCA cycle. Okay, and normally when we're doing work, the NADH is depleted. There are a few situations where NADH production can cause problems with the TCA cycle, particularly with ethanol toxicity. So with ethanol toxicity, generally, like if you're falling down drunk, you're not actually going to be doing a lot of work and driving the TCA cycle or pulling the TCA cycle along. The metabolism of ethanol, both the ethanol dehydrogenase step and the acetaldehyde dehydrogenase step, those both generate NADH. Okay, so it's a fuel oxidation pathway to convert ethanol to acetyl-CoA. Now this acetyl-CoA, we could add that into the TCA cycle and oxidize it down to carbon dioxide. But the problem is that this abundant NADH, this is inhibiting every reaction for which NADH is a product in the TCA cycle. Does that make sense? Okay. So this can result in hypoglycemia as one of the consequences of ethanol toxicity because gluconeogenesis is reliant on substrates that have to go through the TCA cycle in order to be gluconeogenic substrates. Okay? So this is one situation where that NADH is driving the activity of, or preventing the activity of the TCA cycle, not through any allosteric regulation, but just through manipulating the concentrations of the products of the redox reactions. With fatty acid beta oxidation, this we could consider this, say, in a case where we're in starvation metabolism. Okay, so this is, this isn't like a pathological thing, I guess, unless you die from starvation. It's like a normal response to starvation. Okay, so the fatty acid beta oxidation, we know the products of that are acetyl-CoA, and we also get NADH. And we can get a lot of NADH because, each fatty acid, you get multiple rounds of the beta oxidation. So this is just a simplified view of that. Now that in the context of starvation, you're going to want to not burn carbohydrate, of course. The NADH produced from beta oxidation, that's going to prevent the acetyl-CoA from being oxidized in the TCA cycle because of product inhibition of all those NADH generating enzymes in the TCA cycle. Okay, so NADH is going to be inhibitory towards the TCA cycle. If you recall, one of the normal responses to starvation is ketone body synthesis. And that NADH, that can help to promote the ketone body synthesis by being a substrate for the enzymes that catalyze the reduction of acetoacetate to beta hydroxybutyrate. So remember, this is the most abundant of the ketone bodies circulate. Yes?
Why does
I'm sorry, I won't point at you with
why
this.
doesn't ethanol poisoning drive NADH into ketone bodies?
It can.
Okay.
Yeah, so no, I just didn't indicate it here. But yeah, you can get ketoacidosis from ethanol poisoning as well because this acetyl-CoA doesn't have any metabolic fates, or it can't be completely oxidized. Now, one of the things that happens with, or one of the differences, I guess, between starvation and ethanol metabolism, in ethanol toxicity, you get this acetyl-CoA. This is able to become citrates through the citrate synthase reaction. That then is used as a substrate for fatty acid synthesis. So you get, elevated fat production in the liver, and fatty liver can be one of the consequences of chronic alcohol consumption. So I think the metabolic fates of acetyl-CoA are a little bit different here. Whereas in starvation, the body wants to make those ketone bodies physiologically. Okay. Does that make sense? Are there other questions? Okay. Okay. I think I've got clicker slides interspersed fairly regularly throughout this hour, so be ready. All right, everybody have a response. All right, isocitrate dehydrogenase. That's the rate limiting step, and it's the key regulatory step. Okay. So you probably know what's coming next, right? How it's regulated. So the regulation, the activity of the isocitrate dehydrogenase is allosterically regulated by multiple things: magnesium, calcium, and as shown here, the nucleotide adenosine diphosphate. So shown here, this is the relationship between enzyme velocity and substrate concentration in the absence of ADP and then in the presence of ADP. Okay, so rather than just remember or memorize this, I have a few questions. And how comfortable are you with this material? Do we need to have a discussion of this, or would you rather review this on your own? And let's just do it. So I'm trying to have a few different questions that aren't just spit out that it's an allosteric activator, but actually like to be able to interpret the graph a little bit more critically. So go ahead and talk with your neighbor on these two. So. Okay, do we have a consensus? Should see how we're doing here. Very good. Yeah, I think that C is the best answer. Right. So A, definitely not true. B, where's infinite substrate concentration? That's over here somewhere, or in infinite direction that way, but that's the asymptote that this line is approaching, right? And that doesn't change. Okay, so let's look at, same diagram, but different sentence below. What would be the best way to describe this? Okay, it's going to be a 100% one. Dynamite, very good. Okay, so isocitrate dehydrogenase, this is allosterically activated by ADP, and we can, so I have shown here that it's decreasing the Km, just, for a little detail there. Sometimes that's referred to as the S.5 for enzymes that have multiple subunits and have this sigmoidal curve. So I would refer to it as a Km still, just for simplicity, but sometimes you'll see it as an S05. Okay, one more. Whoops. Okay, and I've got kind of two questions, one asking you a little bit to say what the graph is showing you, and then another to say what citrate is doing. So let's see, this is a Lineweaver-Burke plot. Remember the double reciprocal plot of the Michaelis data? And this is line 1 is without citrate, and line 2 is with citrate. And yeah, go ahead and chat with your neighbor if that's helpful. Okay, we ready?
But no, but it's a ratio, so that's
Okay, super. All right, I'm gonna just skip over this one. I'll give it to you after class, but I think we're good on this one, right? Whoops, can you see that? I've got the text covered up there, but so citrate is an activator of isocitrate dehydrogenase, which is 2 steps down the line in the TCA cycle from where citrate is. So what type of regulation would that be? Have you heard these terms before? Okay, good. Okay, so this is feed-forward activation. You've got abundant substrates. You want to increase the rate of the whole pathway. Remember, amino acids all have a keto acid partner that results from their transamination. Remember that? You remember this one? Christine? Okay, good consensus on the correct answer. Aspartate is the transamination partner of oxaloacetate, and glutamate is the transamination partner of alpha-ketoglutarate. Okay. So next question, this is going to come out of nowhere. No right answer for this one. Has anyone been on any, what, have you been on one of these?
Navy Pier 1.
Okay, yeah, that's the one I've been on. Anyone been on any of the other ones? London Eye or Coney Island one? You been on London Eye? Okay, neat. Okay, so should we? I don't know. I guess I mean the big O has a great name, I guess, but I don't know. I like the Navy Pier one was good. Okay, so why do I ask this? So this is a leading question, and it's getting to a metaphor about the TCA cycle as a Ferris wheel, and what do you call the carts on a Ferris wheel? Like the gondola or the seat, or can we just call them seats? Okay. All right, so let's consider the fuel oxidation function of the TCA cycle. Okay, oxaloacetate is the seat, and the passengers that it takes are 2 carbons from acetyl-CoA to make citrate, and then it dumps those passengers off as CO2, and there's another seat of oxaloacetate. Okay, so the capacity of the TCA cycle is how many molecules of oxaloacetate there are, right? That's how much fuel oxidation you can do at any one time. How many seats there are on the Ferris wheel, right? Okay, so the issue here is that the TCA cycle doesn't only function in this fuel oxidation pathway. Okay, so the TCA cycle also has anabolic functions, And two of those anabolic functions we just saw in the clicker slide. We can make aspartate from oxaloacetate if we need that amino acid. We can also make glutamate from from alpha-ketoglutarate if we need that amino acid. So that's two reactions where we can take away seats from our Ferris wheel, right? And there's numerous other ones. Yes?
I looked it up. It's called a capsule.
What is the capsule?
Seat on a Ferris wheel.
Oh, okay, a capsule. Okay. All right. So when things go out of the TCA cycle doesn't have enough capsules, or it doesn't have as many capsules, right? So essentially, what we're going to discuss are anaplerotic reactions or filling up reactions that can, if things go out of the TCA cycle for biosynthetic reactions, these are ways that when the cell is going to switch to needing energy into doing fuel oxidation, these are ways that we can get more oxaloacetate, more capsules in our Ferris wheel, and that increases our fuel oxidation capacity. Does that conceptually make sense? Okay. Okay, so the way that things can divert out of the TCA cycle, are diverse. We mentioned one, the conversion of oxaloacetate to aspartate, and then you can get a few other amino acids from that. And alpha-ketoglutarate can make glutamate, and we can make glutamine and other amino acids from that. Citrate, this is a major substrate for fatty acid synthesis. So citrate can leave the mitochondria and make acetyl-CoA in the cytoplasm, and that's a substrate for fatty acid synthesis. And then succinyl-CoA, this is a heme synthesis substrate, and we'll talk about that in the IHO course. So there's a lot of ways that we can get rid of capsules or get rid of oxaloacetate, and there are also several ways that we can replenish the TCA cycle to increase the fuel oxidation capacity. Okay, so just like oxaloacetate can leave as aspartate in the context of needing more energy generated capacity, aspartate can become oxaloacetate. Likewise, glutamine can be deaminated and glutamate can be transaminated to alpha-ketoglutarate. I think a lot of folks got a question wrong on propionyl-CoA and how that gets converted into succinyl-CoA on the last test. Remember, it gets carboxylated and needs biotin. And then there's the methyl transfer, methyl rearrangement that needs adenosylcobalamin. Okay, anyway, that's down here, just for a reminder. Okay, so let's talk about in a little bit detail some of these different reactions. Okay, so and conceptually, it's because things leave the TCA cycle, we have to be filling up the TCA cycle. Yes?
Sorry, I just have a general question. So are these anaplerotic reactions happening more to like replenish the cycle when you're in like a high energy state, or is it when you have like an excess of the TCA that it like backfills into like its reserves?
Yeah, so when you're in a high energy state, generally things are going to be leaving the TCA cycle and doing biosynthetic reactions. And then when you don't have abundant energy within the cell, if you increase the workload or decrease the nutrient load, then you can switch to the anaplerotic reactions to increase your fuel oxidation capacity. Does that? Okay. There's some, of course, there's exceptions to this. Like for fatty acid synthesis, you have to dip down in here and go to citrate. And for gluconeogenesis, you have to dip down into oxaloacetate and to malate. So there are some like little truncated TCA cycle pathways that aren't so related to that. Okay, let's see. All right, so let's talk about these one by one. The pyruvate carboxylase reaction, this is an important one. It's an important anaplerotic reaction. It's also an important gluconeogenic reaction. And the rest of the details on gluconeogenesis we'll talk about later in the course. So the pyruvate, we can get that into the TCA cycle as acetyl-CoA going through pyruvate dehydrogenase, right? We can also have it go into the TCA cycle as an anaplerotic reaction by using a biotin-dependent carboxylation reaction to make oxaloacetate. Okay, so we could imagine, if you have abundant pyruvate around and you needed to make energy, one pyruvate could go to acetyl-CoA, the other pyruvate can go to oxaloacetate, then they can both come together to make citrate and on in the TCA cycle. Okay, so that's one way, shown here, that you could increase TCA cycle capacity in response to having a lot of pyruvate around. One other anaplerotic reaction, and this is a somewhat complicated one, but it's there's a pathology associated with it called myoadenylate deaminase deficiency. This is a muscle-specific way that we can use amino acid catabolism and link that to work being done to make anaplerotic substrates. So it starts with work being done. So we hydrolyze ATP to ADP, and then the adenosine or ADP kinase reaction can make AMP. So AMP, this can be deaminated to inosine monophosphate. The inosine monophosphate then, this can take a succinyl molecule from, or it can be bound to the amino acid aspartate that makes adenyl succinate. Fumarate is cleaved off of this to regenerate AMP. So essentially what the cycle is doing is it's taking this signal of low energy, the AMP, and using that as a way to get amino acid carbon into the TCA cycle as a way to increase ultimately the amount of oxaloacetate in the cycle. Okay, so just to zoom in on that adenylosuccinate, that is the adenosine nucleotide, but with that aspartate-derived carbon portion bonded to the base portion. And then when you cleave this off, you're cleaving it off. So this nitrogen, this would come from the amino acid aspartate. When you cleave that off, you're generating fumarate. Okay, and AMP. All right, so myoadenylate deaminase deficiency, that's a deficiency in the muscle-specific isoform of the enzyme that makes the inosine monophosphate. And people with this condition, the hallmark symptom is exercise intolerance or pain when you do strenuous exercise. So when you do strenuous exercise, there's a normal physiological increase in ammonia, and it's not hyperammonemia, but it's an increase within normal physiological range. And patients with the myoadenylate deaminase deficiency, they won't have that increase in ammonia. So essentially, the muscle in people with this condition, it can't respond to increased workload by increasing fuel oxidation capacity. The cell then has, the muscle cells have energy starvation, and they compensate for it by increasing glycolysis, and that can cause acidification of the tissue and cause that pain. Okay. I'm going to just leave this with you to go over, because I just in view of the time, I want to make sure that we touch on a couple other topics. Okay. So we're going to leave the anaplerosis and have a discussion on TCA cycle intermediates and how they affect the nucleus. So kind of some interesting links between the two. So did Dr. Cormier talk about epigenetics then? The specifics of it? Okay, very good. So yeah, I think, exon activation and mosaicism, I think they're linked in there somehow, right? But I think epigenetics is the best answer here. Okay, so let's look at how the TCA cycle can affect nuclear gene expression. So there's two things I'm going to talk about. One is substrates or intermediates of the TCA cycle influencing epigenetic gene regulation in the nucleus. Then there's a related concept of fumarate hydratase having a specific role in DNA repair. Okay, so kind of two concepts. Okay, so epigenetics, generally that's referring to reversible modifications of histones and of DNA bases. And the major base is cytosine, and methylation of cytosine is involved in, or is a way to regulate gene expression through epigenetics. Okay, so let's kind of take these one by one. The simple one is the histone acetylation. So acetyl-CoA, this is a substrate for the TCA cycle. So that's our mitochondrial pool of acetyl-CoA. However, when that mitochondrial pool of acetyl-CoA is big, we can generate mitochondrial citrate that can leave the mitochondria through the citrate transporter. Then the acetyl-CoA lyase reaction generates a cytosolic pool of acetyl-CoA, and this can be a substrate for the acetyltransferases that will modify the histone acetyl code. Okay, so that we'll talk in more detail about this step and generating that cytosolic acetyl-CoA pool when we talk about fatty acid synthesis in a couple weeks. Okay, so this has a role in fatty acid synthesis, but then it also has a role in the epigenetics. Okay, so that's fairly straightforward, and I won't spend a lot of time talking about that. The more, I guess, a little bit difficult one is with the methylation reactions. So the methylation reactions, the methyltransferases are going to be using S-adenosylmethionine as a substrate to add methyls for both methylation of lysines on histones and methylation of cytosines on DNA. Okay, so the methyltransferase step, that is requiring folate metabolism and S-adenosylmethionine. Okay, and that's kind of not shown here. The intersection between the TCA cycle and methylation of lysines on histones and cytosines on DNA is with the demethylation step. So the demethylases, or one of the important families of demethylases, use alpha-ketoglutarate as a substrate, and they generate succinate. Okay, so it's like it's using the alpha-ketoglutarate as an electron donor for the reaction to remove that methylation. So when we have abundant alpha-ketoglutarate around, that is going to promote demethylation of the cytosines and the histone lysines. When we have some of these other normal products of the TCA cycle that are dicarboxylates like alpha-ketoglutarate, they're structurally similar enough to alpha-ketoglutarate that they actually act as inhibitors of the dioxygenases that will demethylate cytosines and lysines. So if you have abundant succinate or abundant fumarate around, that can lead to hypermethylation of your lysines on the histones and the cytosines on the DNA, and that can change the gene expression pattern within the cell. Okay, this guy, 2-hydroxyglutarate, this is an oncometabolite, and this is made by mutations or gain-of-function mutations in the NADP-dependent isocitrate dehydrogenases, the IDH1 and IDH2. Okay, but so when this is made, this functions like succinate and like fumarate to inhibit the demethylases. Okay, with me so far? Any questions on this, or is this comporting well with Dr. Cormier's discussion on epigenetics? I don't want to, yes.
So are you saying that the L2HG, that it only builds up if there's an IDH1 or IDH2?
That is correct.
Okay.
Yes, I'll show how that happens in I think just a couple of few slides here. Okay, so this kind of shows, the general cartoon sense of what's happening. This shows really the same thing, but in a little bit more detail. So really the effect here is that we are changing gene expression patterns within the cell. And, that's going to be different for any particular gene or any particular cell context, but just consider it, in a cartoony sense, it's changing from one gene expression pattern to another. Okay, so let's look at those lysine amino acids in the histones. So they can be methylated through the methyltransferase, lysine methyltransferases. These are using S-adenosylmethionine, and we can get either mono or dimethylation or trimethylation. And that methylation pattern, as well as the acetylation pattern, if this is a histone and this is the N-terminal tail, the acetylation and methylation of that lysine make a code that is recognized by the transcriptional apparatus and can determine whether that gene is going to be expressed or not expressed. Okay. So this is a common analogy that is used in epigenetics. There are enzymes that write the code, so that would be the methyltransferases. Then the RNA polymerase II complex, ultimately that's going to be reading the code. And then there are erasers, which are the demethylases. And this is what we'll talk about related to the TCA cycle. So there is an oxidative demethylation pattern shown here. So if this is our lysine and this is the methyl group, this is the oxidative pathway of demethylation. And it generates formaldehyde as a product, so this is one of those rare reactions that make formaldehyde in the body. So the carbon on the methyl group leaves here. So this pattern does not depend on the alpha-ketoglutarate. There are a family of alpha-ketoglutarate-dependent dioxygenases, and they serve diverse functions within the cell, including collagen maturation and oxygen sensing and methylation, as shown here, of lysines. These are using alpha-ketoglutarate as a substrate, and they decarboxylate alpha-ketoglutarate to succinate in a reaction somewhat redolent of the alpha-ketoglutarate dehydrogenase reaction, except they're splitting the oxygen and adding an alcohol to the methyl group. So this alcohol methyl then, this gets cleaved off as the same product, formaldehyde, and we get a demethylated lysine. Okay, so this is, so it's this pathway that is going to be impacted by the intermediates of the TCA cycle. Okay, so this is essentially, excuse me, the problems can occur if you have mutations in TCA cycle enzymes that would generate too much, that would prevent the complete oxidation of the, that would prevent the cycle from going back to oxaloacetate. So if you have a mutation in succinyl-CoA dehydrogenase or in fumarate hydratase, you're going to build up succinate and fumarate, and then those are going to leak out and promote the hypermethylation of your lysines, and that's going to change the transcriptional pattern within the cell. So mutations in those TCA cycle enzymes, those can be oncogenic by generating excess succinate or excess fumarate. There are also mutations that are oncogenic not in the TCA cycle, but in the two non-TCA cycle enzymes that catalyze the isocitrate dehydrogenase reaction. So there are gain-of-function mutations in IDH1 and also in IDH2 that can have the enzyme work in reverse, and instead of making alpha-ketoglutarate, they're taking alpha-ketoglutarate and turning it into hydroxyglutamate. Okay, so this is, rather than generating alpha-ketoglutarate, they're depleting alpha-ketoglutarate, and they're making this product that isn't made anywhere else in the cell, but can act as a potent inhibitor of any of the alpha-ketoglutarate dependent dioxygenase enzymes, such as the lysine demethylases. Okay, so this would be the normal IDH1, IDH2, and then the mutant one is taking alpha-ketoglutarate and making the oncometabolite hydroxyglutamate. So you can have a mutation in one or the other, right? So if you have a mutation in IDH1, then IDH2 is going to be generating alpha-ketoglutarate, and the mutant IDH1 is going to be making the oncometabolite. If you have a mutation in IDH2, then IDH1 is going to be doing this, and the other one is going to be doing that. Anyway, there are mutations in both that can generate that oncometabolite. Okay, and the significance is that 2-hydroxyglutamate acts as a competitive inhibitor for all of the alpha-ketoglutarate dependent dioxygenases. Okay, and the consequences here can be significant. So the IDH1 and 2, those are actually considered proto-oncogenes, and when they gain these gain-of-function mutations, that can promote cancer and the accumulation of additional mutations because it changes the transcriptional pattern of the cell to prevent differentiation and promote proliferation. Okay, and the more proliferative the cell is, the more it's going to accumulate other additional mutations that can result in a pathological cancer. Yeah.
So they're not activated in the cytosol until there's like that buildup of like this, and then they become oncogenic?
No, well, there are two ways that you can have this. So the fumarate hydratase in the TCA cycle, that's acting as a tumor suppressor. If you have a loss of function mutation in fumarate hydratase, you're going to get fumarate building up. That's going to be then promoting the hypermethylation of the cytosines and lysines and promotes that oncogenic gene transcription pattern. IDH1 and 2, those are acting as oncogenes. So those need to become activated or have a gain-of-function mutation to generate the oncometabolite. You talked about tumor suppressors versus oncogenes? A little bit? Okay. I want to get through this material, so let's maybe put a pin in that. Okay. Okay. Anyway, so you can get this, you can get the stuff up above here leading to tumorigenesis either through a gain-of-function mutation in IDH1 or 2, or to make the 2-hydroxyglutamate, or a loss-of-function mutation in fumarase that would lead to a buildup of the normal intermediates in the TCA cycle, which would function like 2-hydroxyglutamate. Okay. Okay. So in addition to impacting gene expression through epigenetics, these alpha-ketoglutarate dependent dioxygenases have other functions as well. And one of the functions is in oxygen sensing through the hypoxia-inducible factor. So the normal pathway in the presence of oxygen is this hypoxia-inducible factor. This has proline residues or proline amino acids that can get hydroxylated by an alpha-ketoglutarate dependent proline hydroxylated. So these prolines get oxygen added in the presence of molecular oxygen and alpha-ketoglutarate. And then the products are succinates and carbon dioxide. The function is, as an oxygen sensor, is these prolines, when they're hydroxylated, they are recognized by the von Hippel-Lindau factor, and that leads to ubiquitination and degradation of the protein. Okay, so in the absence, or in the presence of oxygen, this protein gets made, hydroxylated, ubiquitinated, and destroyed. When oxygen is absent, then the substrate is gone, and the hypoxia-inducible factor goes to the nucleus, and it causes a transcriptional pattern to change. And what it's trying to do there is to compensate for the loss of oxygen. So you're going to get increased glycolysis, because that's an energy-generating pathway that doesn't depend on oxygen. And you're also going to get increased angiogenesis. Angiogenesis is making new blood vessels that would bring new blood and new oxygen to the tissue. Okay, so both of those things, they're a good thing to happen if you have a poorly perfused tissue, for sure, but those are also things that are necessary for a solid tumor to grow. Right, recall the Warburg effect, or that increased glycolytic rate of tumors. And tumors also, solid tumors need to recruit vasculature. Otherwise, they're going to be dependent just on diffusion to get nutrients and oxygen. So essentially, if you have 2-hydroxyglutarate, for example, or abundant succinate or fumarate around, that's going to set up a pseudo-hypoxic response in the cell. Okay, even if there's plenty of oxygen around, the oncometabolite is going to be competing with alpha-ketoglutarate, and you're going to go down this pathway. Good? Okay. Okay, I'm going to just beat this to death. But this is something that we've seen before, I think, with the vitamin C dependent prolyl and lysine hydroxylases that are important for collagen maturation. So these are also alpha-ketoglutarate dependent dioxygenases. If they're deregulated in the presence of an oncometabolite, that can derange the extracellular matrix, and that's something that cancer cells need to be invasive and invade into healthy tissues. Okay, so just to summarize here, we've got cytosine demethylases, chromatin or lysine demethylases for chromatin, oxygen sensing, and then extracellular matrix. So there's all diverse functions of these dioxygenases, and they're all impacted by the TCA cycle intermediates because they all require alpha-ketoglutarate as a substrate. Okay. Should I do one more topic, or should we just, why don't we just break there, and if anybody has any questions, I'm happy to answer. I know we've gone through a lot here today. I'll cover the last little bit of what I wanted to do today tomorrow, but it's going to be more of the same stuff. I'm warning you. So there's a interesting action of fumarate hydratase in the nucleus, and that is generating a inhibitor of the demethylation, again, of lysines, and that's important for DNA repair. So it's going to be familiar to you. Okay, anything else? We've got one more minute. I feel like I ask if there's any questions, and then I don't give you sufficient time to think of them. I'll say, any questions? No? Okay, let's move on. And I kind of, it's a habit that I have. I know it's not good, and I'll try to break it. But if you do have questions, just raise your hand and get you. Okay, well, hearing none, it's 9:50. Let's do other stuff.
Like how, what expectations?
I had it on my calendar the other day, and then I have it on my calendar today. I'm just scared. I know it's like all those photos, but I'm just like, I know. Oh yeah, okay, we'll get pictures. There's like 3. Oh, really? Alright, go for it. I'll be here. Yeah. Who was it? So, do you have like an idea?
Dr. Stover something.
Oh, that was always me in Run Club too at the U. I would like meet people and I would like forget their name and then we would go running together for like multiple times. Yeah. How fast were you guys? It's comfortable. Okay.
My problem is that I probably already the next calendar.
I also just like the motivation. It's like, well, you know, I can show up, and then I don't actually mind running by myself. But then I meet someone, and like, it's just hard. That was like always my favorite too. And why it was hard, like if you stop going, you know, or like need to take a break to like get back into it. Yeah, because I'm hard. I do want to like.
Not that I want to do that, but
You know, like second time I'll go there first. Meet the other people who enjoy running and stuff like that. So we'll see if I work up the nerve. Yeah. Yeah. Well, have a good rest of your day. Thank you. We'll see you later. Yeah. We'll see.
Yeah, I would see your cycle. Was it a pretty general consensus? No, like it was like one of the
April,
same.
let me know when you come back.
I will. It must be like defined.
Yeah. Alright. Now I'm like, oh crap, now I need to go over this lecture because I have no idea what just happened in the second half of that lecture. I know, it's like, the second half always gets me. I know, that's why I was just like,
This is cool for that.
I'll text you. Okay, sounds good.
I've been trying to put a little notability category for something fun that I learned.
Oh, cute.
That's what I'm going to put in there today.
Capsules.
Definitely not going to write about what kind of TCA cycle.
Humor and all these dehydrogenases, whatever.
Like the middle one has a bunch of axes. You know, like Taz, Ax. Those are the 3rd gen.
Okay, I like it. I need that. That's what I need. I should go.
The cephalosporins. Those are the hard ones because they're all the same names. They're all Ceph. The other ones are a little bit more different.
I need to go look at them. I really haven't. I watched the lectures and then I was like, another day.
Yeah, like, oh.
Yeah. So I gotta work through.
Some of them are nice because it's just one. It's just.
I laughed so hard at your 70% best friend.
So you're like, I hope for 60%.
Like, I'll aim a little bit
I was
higher.
like, just like a little bit. I was like, just do it. I just opened up your snap. Wasn't that funny? Oh, I hope that you get the 70%.
I know. I mean, I told him, I was like, Mom's like, all these extra plants, you should take some because it's supposed to be winter. And I was like, Mom, only if you like, you literally can't kill them.
You're like,
Like,
no,
if I-
seriously.
If I could do more than like water them like once every week.
How many do you
then I
have?
know. I have like 9.
What did you plant, Dad?
I probably had like 20.
Oh.
But slowly they die off, and then the really hardy ones that are really hard to kill just stay.
Do you have any like succulents, or is that?
They're mostly succulents.